Thank you. So I'm going to speak about the genetics of brain structure and function and about 3,144 GWAS in the UK Biobank. And I'm going to start giving a brief introduction on the UK Biobank project. So for all of those of you who don't know, Biobank is a cohort study with 500,000 participants, aged 40 to 69 years old from the United Kingdom. And we have gathered biological samples, questionnaires, and physical examination from all these subjects. Um, um, we have characterized this all the participants before having any health outcomes, and it's important to note that this sorry that this is a prospective epidemiological study, so it's not focused on just one disease. Um, uh, we have now acquiring images for 100,000 subjects. The modalities that we are acquiring that are not brain are, among other, cardiacs, abdomen, or bone density. And for brain imaging, we are acquiring 6 different modalities. So far, we have acquired images for 25,000 subjects. And in the genetics part of the project, we have acquired the genetic information of all the participants, the 500,000 subjects. The original array is 820,000 SNPs, but this has been imputed to 96 million genetic variants. And all this data was released last July, and it has been used, for example, in the Enigma project. This resource is available for every group that wants to use the data. But you have to follow an application process where you have to state the scientific question you want to ask. And this process is needed in order to protect the privacy of the subjects. There's a small fee of £2,000 that is needed to maintain the resources in the long term. But this fee is smaller for low-income countries. And we want to make clear that we don't have preferential access to the data. We had to follow the same application process than the rest of the groups. And that is the link to access to fill this application for the data. So about the brain imaging, in the structural modalities, we have T1 weighted, T2 flare, and susceptibility weighted images. In diffusion, we have 100 directions into cells. For task fMRI, we have a protocol to find activation in the visual cortex and in the amygdala. And we also have resting state fMRI. And with the images from the raw icons, these images go through an automated processing pipeline based on FSL. And out of that, we get a set of process NIFTs and what we call IDPs, imaging-derived phenotypes, which are basically summary measures that describe a certain feature of the brain, like the cortical thickness, or in this case, the volume of the left thalamus. And what we can do with these IDPs is correlate them with other imaging variables or non-imaging variables. And for example, here we are showing a Manhattan plot with 40 million univariate associations that we did between IDPs and non-imaging variables. And as we are saying there, 15,000 of these associations have a significant value higher than the Bonferroni threshold. And about the genetic part of the project, we started with 8,400 subjects with a common genetic ancestry. From the initial set of 96 million genetic variants, we just kept 11.7 million SNPs, applying several filters. And we ran different GWAS per IDP, as we have 3,144 IDPs, including free surface volumes and subcortical volumes of many kinds. We ran 1 GWAS per IDP. We found 148 significantly associated clusters, and these results were replicated in a different set of 3,000 subjects. And the Manhattan plot that you see in the bottom is one example of what kind of results we get out of the GWAS. And you can see, for example, in this Manhattan plot, 4 different SNPs that are significantly associated with this IDP. And we can find spatial relationships between these SNPs and the brain. For example, if we run a simple GLM analysis using the image that is associated with that IDP and setting the absence or presence of the SNP for each subject, we can get for each SNP, we can, for example, in this case, this GWAS is the mean intensity in T2 star in the putamen. So you can see that each different SNP is affecting the putamen in a different region, and that's an interesting result. And all these Manhattan plots and the spatial maps are available in the Biobank Big Imaging Genetics server. And I'm including here 2 examples of the many that we found. This is the tensor mount in the crossing-pontine tract. And first we found a hit, a GWAS hit in chromosome 11 with a p-value of 10 to minus 18.8. But in the same cluster, we found another hit. And if we represent those 2 SNPs, the spatial maps that we found with those 2 SNPs, you can see the blue and the red in that image. It's affecting a very concrete part of the brain, and those SNPs are also related with those 2 genes, CMA3D and ROBO3, which are also affecting axons during development. And these genes are also known to be related with horizontal gaze palsy, which is a disease that is known to be related to those crossing tracts. Finally, the last GWAS that I wanted to show is the intracellular volume fraction cingulum, which with one significant hit in chromosome 5 with a p-value of 10 to minus 30. And if we get the spatial map out of this SNP, you can see that the effect is widespread across the whole white matter. And that SNP is related with genes associated with the extracellular matrix. And also, these genes are known to be associated with plasticity and with myelin repair, and with diseases like depression, ALS, and MS. Finally, the take-home message is that with UK Biobank, you can get the relationship between the genetics and how each SNP is affecting the brain, and also in more general way, the general relationship between diseases, genetics, and brain imaging. That's all.
So we